All right. Good morning, everyone. Thank you. Thank you today for coming to our quarterly series of Biotech Connector. It's great to see a lot of folks here in person, and I know we have a lot of folks online as well. My name is Vladimir Popov. I'm a Chief Innovation Officer here at the Frederick National Lab. And today, the Biotech Connector, we'll be talking about antibodies, as you guys can see. Very exciting topic. I'm going to cover a little bit about speakers in a minute. But just to tell you what, for those folks who are attending this for the first time, Biotech Connector, it's a collaborative effort between Frederick National Lab and the Frederick County Chamber of Commerce. We've been doing this for quite a few years. We've done it through pandemic, switched to online, came back to in person. Every time we do this, it's a quarterly speaker series. We do a topic-specific event where we bring speakers from the region that are experts, and then we also bring one of our own. So a little bit about our Frederick National Lab. Frederick National Lab is a national lab. It's located right here in Frederick, Maryland. We're a collaborative enterprise. We, as a national lab, we're a national resource, so we heavily collaborate and partner. But for those who have companies and who are also looking for business, we do a lot of subcontracting, so there are business opportunities online on our website. And there are also job opportunities here all the time. We're an enterprise of about 2,400 people, so you can imagine that there are job opportunities as well. For collaborative inquiries and questions, we have members of our partnership development team. We have Amanda, we have Victoria, Luba. So feel free to talk to them, talk to me after the event during the networking. So I mentioned that we have an amazing event focused on characterization and various application of antibodies. But before we get into our speakers and their backgrounds, I just wanted to go through a little bit of logistics. We'll move through the speakers. You can see the speakers on screen, and we'll have time for a question or two. If you have a question, please stand up. You can state your name and where you're coming from and ask the question. For online folks, you guys don't have to stand up unless you want to stretch. You can either unmute yourself or you can post a question in the chat, and we have someone monitoring the chat, and we'll read the questions. Also, one detail, unlike most of our Biotech Connector events, we have two speakers are online today. We usually get all the speakers in here, but that shouldn't make a difference. You will still enjoy their presentations. So they're more than willing to stay longer online and answer any questions that you might have. Dr. Simona Colantonio, who's sitting right here. She's one of our own. She's in person, so you can approach her after a presentation or during networking. So let's move into the speakers. And today I have a pleasure of introducing three amazing speakers in this field of research. Dr. Simona Colantonio is a director of Antibody Characterization Lab, ACL, here at the Frederick National Lab for Cancer Research. Dr. Colantonio is an expert in mass spectrometry applied to biologically relevant molecules. She has obtained a master's and doctorate of pharmacy degree in pharmaceutical chemistry and technology from La Sapienza University in Rome, Italy. Did I say that right? Okay. And later she received her Ph.D. in pharmaceutical sciences from the same institution. In the early stages of her career, she worked at the Regina Elena National Cancer Institute in Rome, Italy, where her studies focused on pharmacokinetics and anti-cancer drugs. After moving to Maryland, Dr. Colantonio became involved in serum biomarker discovery while working on a postdoctoral fellow at the formerly SAIC Frederick, now Frederick National Lab for Cancer Research. Over the years, she has gained a deep knowledge in antibody-antigen interaction and has become familiar with growing numbers of antibody characterization tools available at the ACL. Dr. Colantonio has been serving as ACL director since September of 2019. Our second speaker, Dr. Won-Jin Ho, is an assistant professor of oncology and director of mass cytometry facility at the Sidney Kimmel Comprehensive Cancer Center at Johns Hopkins University. Dr. Ho focuses on cancer, immunology, and tumor microenvironment research in gastrointestinal cancers. He leads NIH-funded translational efforts to employ emerging high-parameter tools to empower biomarker discovery and development of novel immunotherapeutic strategies against cancer. He has received awards from ASCO, ACR, DoD, and Harrington Discovery Institute for his work in cancer immunology. And last but not least, our third speaker, Dr. Monirath, is the Director of Pathology and MacroGenics. Dr. Hov is a translational research pathologist focusing studies on cancer immune microenvironment using multiplex tissue-based imaging platforms, as well as biomarker discovery for drug development. She has received an MD degree from the University of Health Sciences and has completed residency at Ghent University, where she later received a Ph.D. degree in pathology. So please grab coffee if you haven't done so, grab some food, and enjoy the talks. First speaker will be Dr. Colantonio.
Thank you, Vlad. Thank you. And good morning, everybody. Thank you for being here. And thank you to the organizer to give me the chance to talk about some of the work that we do here at the Frederick National Lab. So my name is Simona Colantonio. I am the director of the Antibody Characterization Lab. So we work with research antibody. The research antibody market is a pretty large market and is destined to grow in the next few years. From this study that I found from June 2023, so it's a pretty recent study, we know that basically 3/4 of the market is taken by primary antibodies. This is not surprise. The primary antibodies are the antibodies that directly detect the target, the molecule that we are interested in. Most of the time, this is a protein. 50% of the market is Western blot. This is another big surprise. We'll talk about Western blot because obviously we do that. 1/3 is oncology, and this is again not a surprise. There is much interest in this field. And by the end user, the majority of the end user are actually academics and research institutes like us, basically. So we do spend a lot of time and a lot of money with antibodies. The problem is that not always the antibody that we can get are up to the challenge that we need. So this is a big problem in science. This is known for a while. And the problem that I have with this is that if we rely on the wrong antibody, what happens is that we will obtain misleading results that inevitably will bring like a waste of time and resources. But this, for me, is bad science. They will cause an indirect harm to the patients because it will delay the finding of really outbreaking solution for them. So in this environment, in 2008, the NCI decided to create the antibody program. And what we do every year or so, we will, sorry, I went too high. So every year or so, we open a call out to the general public, to the scientific community, and we will create fit-for-purpose antibodies. These antibodies have to be obviously related to cancer somehow, because it's an NCI program, and there has to be no antibodies directed to that particular target. If there are antibodies, maybe they do not work in the particular application that they're interested in. At the end of the process, those antibodies will be available to the whole scientific community through the DSHB, the Development and Studies Hybridoma Bank, for a very limited cost. All the data that we obtain at the Antibody Characterization Lab will be posted on the antibody portal, the antibody.cancer.gov. At this point, at this time, we have 943 antibodies developed to 567 different targets. We can search the target by name. We can filter by application type. And also you can see there is a link directly to the DSHB in case we want to purchase that particular antibody. So, like I say, we do a fit-for-purpose development of antibody. That means that we expect for that antibody to at least work in the application that was meant to. But the role of the ACL at this point is to discover if there are more applications for that particular antibody in other fields. So what we try to do is to get like a big picture of the antibody through affinity studies and to a basic functional characterization of the antibody. Once we know the results from the basic functional characterization, we decide if that antibody will be elevated to the next stage of characterization, which is the cell-based and the pathology studies. So I'll go through very quickly. I know it's a lot I'm going to go through. I'm very ambitious, but I really want to highlight everything that we do. So we have 3 different ways to measure the affinity, which is the strength of the binding between the antibody and its antigen. The first method that we use is the surface plasmon resonance, also known as SPR. And what happened in this case, we try to measure the binding that occur between the antigen antibody on the surface of a chip. This will result in a change in mass that can result itself in the reflective index change. We monitor, we can measure this phenomenon, and we can obtain basically information about the kinetic affinity values. The lower is the KD value, the tighter is the binding. Similar way, but different, is the BLI, biolayer interferometry. Again, we are going to measure the binding that occurs on the surface of a device. This time the device is not a chip, but it's a sensor, it's a dip and read kind of device. And again, we can measure the phenomenon of the interferometry, and we are able to study the kinetic and affinity parameters. Another way, more traditional, I should say, is the indirect ELISA. And in this case, the antigen is immobilized on the surface of a plate, and then we go with primary and secondary antibody. We read our plate, and we obtain a value, which is the EC50. Lower is the value, the higher is the strength of the binding. Again, what we do, most importantly also is the basic functional characterization. We have different ways to do that. Just to get a big picture of what we do, we do two different types of Western blot. We do immunoprecipitation, and we do reverse phase protein array, and I'll go through each one of these. So Western blot, you remember, is half of the market of the antibodies. Basically, every lab that does proteomics will have a device to run traditional Western blot. Cheap, reliable. It's very simple. You separate your protein sample on a gel. From the gel, you transfer the protein to a membrane. The membrane will be probed with primary and secondary antibody. We use this to verify that the protein is actually able to recognize the full-length protein in its recombinant version first, and then we try to see if the same antibody is able to recognize the endogenous protein in much more complex substrate, like a tissue lysate, a cell lysate. This is very reliable and very versatile, but we also try to use the automated version of the Western blot. The concept is still the same. We have a separation based on the molecular weight. This time it doesn't occur on a gel, it occurs in a capillary. Then there is a UV laser that irradiates the capillary. This cross-link the protein to the internal wall of the capillary itself. Then we put primary, secondary. What we obtain is an electropherogram that can be visualized as a virtual blot, in other words. These are some, as an example of what kind of data we obtain. And we particularly like this application because we can have a better understanding of the difference in expression level of the same protein across different substrates. Immunoprecipitation is extremely important for very many applications. In this case, we conjugate basically the antibody to the surface of magnetic beads. Then we use those modified beads to probe a protein sample. Everything is handled by a device that is called Kingfisher. And then we will basically wash the unbound eluting in acidic condition, and the eluate can be analyzed. In this case, I put as an example with the simple Western device, but it can be done also in traditional Western blot and also in mass spectrometry. Reverse phase protein array is the last of our basic characterization application, and the layout is similar to an ELISA. We have cell lysate that are applied on the surface of a 96-well plate, probed with primary, secondary, and then we read. What we generally use for this particular application is the collection of cell lines called NCI60. Probably most of you is familiar with this. This is a collection of cell lines that contains group of representative cell lines for different groups. We have ovarian cancer, breast cancer, and so on. So basically, when we do this initial basic functional characterization, the question that we're trying to answer is there any evidence of binding? If the answer is yes, then of course we'll proceed to the advanced characterization. The answer is no, we don't give up easily. So we try again to try different test substrate, whatever is possible, whatever it makes sense. And then if the answer keeps being no, then we have to draw the line because we have to be mindful of our resources and our time. The reason why we don't advance everything to the next phase, which is cell-based and histology, is because cell-based requires cell culture. Cell culture, as you might imagine, is time-consuming, labor-intense. It's a lot going on. So, of course, again, we have to be mindful of our resources. We have to limit this. It doesn't make sense to do it with an antibody that really has no hope to work beyond the application it was created for. And histology, the limitation is the availability of the samples. What we use are tumor microarrays, TMAs, and those are basically thin section of needle biopsies that are applied on an array, and that will be stained and probed. So why we want to advance the characterization wherever possible to this advanced techniques, because they can enable a lot of science. We can learn a lot when we can apply this. For example, single-cell Western blot is a very interesting technique. In this case, we have a suspension of cells, alive cells, that will have to be applied on the surface of a special chip. This chip contains all wells that are big enough to accommodate for one cell and one cell only. With the device, we will basically lyse the cell in the little well, and then we will apply a current to separate the protein content. Imagine this to apply to hundreds of cells at the same time. And what we obtain are numbers. The numbers represent the number of cells that actually express that target and the intensity of the expression of that target. This is just an example of how we can see the data, we can visualize the data, but there are way more ways. I mean, now for the matter of time, I'm going to explain all of them. But what I want to point out, though, is that we always normalize our data to an housekeeping. In this case, is a beta tubulin. Another way that we use basically a still count cells is the flow cytometry. Flow cytometry is extremely important because it's already used in clinics to diagnose certain diseases. And again, we stain our sample, we fix and stain our sample, and then we analyze it. And we always try to use negative control, in this case, the isotope control antibodies, and we also try to use cells that from other applications we know they have different level of expression, so we verify this with the flow cytometry. Another important set of applications are the imaging application, immunofluorescence and immunohistochemistry. For immunofluorescence, also known when applied to cells as immunocytochemistry, we have, of course, a cell culture that we have to fix. Cells will be fixed and blocked, and again, probed with primary and secondary antibody, and we obtain these pretty pictures. We always monitor the nuclei and the microtubules so that we can determine exactly where the protein is expressed inside the cell. This particular case, this protein seems to be expressed at the nuclei level and the cytoplasmic cell level. Immunohistochemistry, I know that you heard about it, you know the importance of immunohistochemistry in clinics. So it is very important for us to determine if our antibodies actually have a potential application in immunohistochemistry. This is just a general layout of the, how we stain our cells. But what happens is basically at the end, we will obtain staining pattern, and our pathologists will go and look at the staining pattern and decide if those make sense basically with the information that we already know from the literature. So this is my conclusion. I know I said a lot of things. I know there is a lot that I've been talking about, but if there is a message that I want you all to bring home today actually, is that we do need better antibodies. And the reason we need better antibodies is because better antibody enables better science. And when you have better science, you will find the faster and better solution for the patient, which is our goal. Now, of course, I need to thank the people that make all this possible. This group of people you see in their name over the presentation, those are the people that make everything possible. They actually work in the lab, or somehow they're affiliated with the ACL. Of course, I need to thank Dr. Hewitt, which is the pathologist that reviews our staining pattern and decide what is worth to call positive, what is worth to call negative. Of course, we need to thank our patron at the Office for Cancer Clinical Proteomics, the NCI, Tara Ilke and Henry Rodriguez. I want to thank also our collaborators, Carla Daniels at the DSHB, and of course, my supervisor, Dr. Niels Lee, that is the head of the Cancer Research Technology Program here at the FNL. If you have any question, I'm here to answer. Yes.
That was a great talk. Of the, you said it was like 950 antibodies you guys have generated and characterized. How many of those have gone through like the full characterization?
Not all of them, because this 950, I don't remember now, but all those we've been collecting them, since 2008. At the beginning, we didn't have all the tools that we have right now, but we are doing now sometimes the effort to going back and try to revisit some of them. We started with simply Western blot, traditional Western blot, and IHC and reverse phase protein array, they were always there from the beginning. We have been adding pieces by pieces. Imaging flow cytometry is very recent. Single cell Western blot is very recent. So we've over the years, this program has been going on for several years. Things were not available at that time. I mean, even 2008, there was simple Western didn't exist, and not even single cell Western blot.
Thank you, Simona. Okay, while we're waiting for Dr. Ho, I wanted to quickly say thank you to our sponsors for the breakfast, McBee Moore and Vanik. And Dr. Ho, you can unmute and start the presentation.
Great, thank you. Can everybody see my screen?
Not yet. Okay, yes, we can see that.
Oh, great. Perfect.
Okay.
Great, thank you so much for inviting me. Just really a pleasure to be here. I wish I could have been there in person. Hopefully there's going to be another opportunity in the future where I can do that. Today I'll be just talking to you about mass cytometry. I try to also include some basic information as well, in case you're not familiar, and also end with sort of a use example. And so a lot of the work that we're doing is in cancer immunology. So hopefully this will be relevant to the work that you're doing as well. So just the overview of the technology. This actually has been around for quite some time. I mean, in 2009 is when it was first developed. Really, the technology is a mass spec machine that's been modified to look at a per cell basis here. So the basis of the technology is utilizing antibodies. We talked about antibodies a lot today, but it's metal conjugated antibodies that can provide the signal readout. And so these cells are then nebulized into single cell droplets. These are introduced into the plasma and you generate these plumes of ions. They get filtered through this quadrupole. And so these common ions that you see in biological specimens, the carbons, nitrogen, oxygens, those get filtered out. And then the probe ions, the heavy metals, are the ones that get measured by time of flight, detected by time of flight. And that's why cytometry by time of flight, or CyTOF. You can use the same technology really on an imaging mode as well. And so imaging by cytometry utilizes the same fundamental technique. Again, antibodies conjugated to metals, heavy metals. And then this instrument then uses UV laser to then ablate the tissue to generate the plumes of isotopes. And so instead of a per cell basis, now you're looking at the metal ion content at a per pixel basis, and each pixel is about a micron squared. So it's still single cell resolution, subcellular resolution. It's not a true optical image as in a microscope, but this per pixel data is then rendered into images that we can analyze. The inherent advantages, as you might expect, are that there are no spillover issues. So we're using heavy metals that are isotopically enriched. So each metal has its own channel. Fluorescence cytometry, as you know, has difficulties with spillover that we have to design very carefully, especially when it gets above 10 markers or so. So that's really allows for high customizability. There's also no issues with autofluorescence. That's often a problem with certain tissue types like liver, brain, bone, or kidney, even lung and pancreas. So really, the signal to noise ratio in those tissues are excellent. We also are quantifying the total ion content. So the readout is more quantitative intrinsically than, say, fluorescence, where there's a certain excitation energy that's also in the calculus there. For imaging specifically, what's also a strength for this technology is that it's compatible with standard histology slides. So if you have archives of tissue slides that you want to use, or standard pathology lab is really the only access you have, and they don't have ability to utilize specialized slides. So this technology would also facilitate that. Just very briefly, the conjugation chemistry, in essence, is taking these, again, isotopically enriched metals, loading them onto polymers. These are chelating polymers that have maleimide moieties, gently reducing the disulfide bonds to have these diol groups. Then by click chemistry, these metals are now covalently tagged onto the antibodies. Currently, there are 50 plus metals that we can use. The blue are the ones that highlight the ones that we can use. For certain noise issues and things like that, 40 are available for imaging. So again, really a variety, and because the conjugation chemistry is highly reproducible, the metals can often be just interchanged without a lot of headaches in designing the panel. These are just some applications that goes beyond what is really a standard or routine use of mass cytometry. Here you're using metal tagged tetramers, so we can screen many different antigen specific T cells. Here you're using an antibody that is cross-reactive to CFSE to get proliferative cells assayed and profiled at the same time using 40 plus targets. Here are using the panels to look for certain metabolic markers to really get at the metabolic profiling of immune cells, which is becoming increasingly important in cancer treatment strategies. And utilizing imaging by cytometry as a 3D platform, so serial sections or full thickness tissues, that's also has been demonstrated. And then also using a version of in situ hybridization to get at both RNA and protein, make use of the 50 channels or so to really understand the biology at different scales, so multi-scale assaying here. Just to compare some of the, again, strengths and weaknesses or pros and cons of the technologies, for high parameter profiling in immunology nowadays. I believe a lot of you will be familiar with oligonucleotide tagged antibodies. So this really depends on sequencing platforms. So having unique oligo sequences to then identify the target. You have a theoretically very high number, really limited by the sequencing depth, essentially, that we can assay at once. Here, there are differences in minimum cells per sample needed at the start of protocol, cell capture rate, and so forth. But I think some of the really key differences are the following. So here, flow cytometry is really the only technology that allows for downstream analysis. So flow sorting for cells, and then doing additional analysis that are guided by the initial markers to sort for. I've mentioned earlier how interchangeable some of these metals are for the different antibody assignments. So very high customizability for mass cytometry. It's not as versatile, I don't think, in the field for fluorescence or oligonucleotide tagged antibodies. Certainly there are a lot of catalog ready antibodies that are out there, but in terms of being able to customize these things, I think mass cytometry is a little bit easier. There's also a lot of pre-processing needs for sequencing data. And so to get the counts matrix and so forth, there's a lot of steps that are needed, perhaps normalization and things like that. And so the readiness of analysis is also different. Mass cytometry is fairly quick to be able to interpret the data and flow, especially 1 to 2, 3 parameters. As you know, you can just use biaxial plots to analyze. So the analyst readiness is also an important factor. And if you take all of this together and also consider the ability to barcode samples with mass cytometry pretty flexibly, you can also bring down the costs quite a lot. And so cost per analyte is a big consideration. So really the field is rapidly changing. I just want to give you just a visualization of what is out there in terms of the spectrum of profiling assays. And this is far from what is comprehensive. Also want to note that this is really not in scale, it's purely for visualization purposes. But you have all these different platforms that give us spatial data, and this is marked in red here. Suspension data, right? Single cell suspension in blue. Got the bulk, of course, that we're all familiar with. Some of these work with protein, right? These indicated by squares on this plot, and then triangles are RNA. So different scales of molecular scales here, different spatial scales as well. So some of these platforms allow for bigger images to be taken at once in smaller regions, and really kind of differentiated by how much it might cost and the dimensionality of these platforms, how many parameters at once. And so you can kind of appreciate that mass cytometry sort of falls in the mid-range of all of these factors. The workflow here is, probably easy to understand because it's very intuitive. It's akin to flow cytometry for CyTOF and akin to immunohistochemistry for IMC. So there's not a lot of learning curve at that level. Panel design, certainly there's a lot of consideration there. So that's probably a time-consuming step to figure out which markers we want for the research question at hand. Do we have the right clones? So earlier we just had a speaker stress that, and that's definitely applies here when we're designing panels. We have the right clones. What are the controls? Can we rely on the data? And then of course, the analysis step, depending on the depth of analysis that you want, it can take a long time. And so really the actual wet lab portion of all of this is quite simple. So moving on to an example of what we have done using mass cytometry. This is just one example. We have many different contexts that we use this in, but just to give you a flavor, so at our Hopkins liver cancer group, we recently ran a trial in the context of hepatocellular carcinoma, looking at immunotherapy, taking advantage of a TKI, tyrosine kinase inhibitor, cabozantinib, targeting a VEGF pathway, and nivolumab is a PD-1 inhibitor for checkpoint immunotherapy. Using this combination in the neoadjuvant setting, so prior to surgery, the goal is to downstage these cancers towards surgical resectability. Otherwise, it wouldn't be a curable effort here. And then these surgically resected samples give us a really nice opportunity because it's ample tissue to interrogate. So we can take these FFPEs and look at bulk RNA-seq Visium or immunohistochemistry, imaging by cytometry, of course, and then the peripheral blood specimens, we can then take them longitudinally and understand what's happening in the peripheral blood using suspension mass cytometry or single-cell RNA-seq. So we have done all of this, integrate all this data to understand the effects of the immunotherapy and perhaps differences between responders and non-responders. This trial in particular was an encouraging trial. This is one of the very first really neoadjuvant trials in the liver cancer space. We had 12 patients, and 5 of them had responded pathologically. So good signal there. And then we were able to then analyze the entirety of the cohort at once. And the way we did that is by generating a tissue microarray. And so you might be familiar with this, but essentially a pathologist can pick out representative areas from a given surgical specimen, and then we can core those areas out and then construct a TMA. And so 3 to 4 representative areas. So we have 37 total cores to represent the entire cohort. And then we can look at this represented image here. It's just a set of 6 markers. And then the responders, generally speaking, you have fewer cancer cells marked by the green ECAD here and there, and then many more immune cells here. I'm marking macrophages. CD68 is showing in yellow. So more immune infiltrated, less cancer in general in the responder core. So already at gross sort of level, we can appreciate the differences. The power of the technology we want to leverage is the ability to then look at it at a per cell basis. So we can segment these images into single cell. And so using a software pipeline, we were able to then take these images and really turn them into a per cell. So these are boundaries around the cell. About 60,000 cells from this entire cohort was exported. And using the single cell data set, we can cluster them as you would for any single cell data. And here's one depiction of that. So flow sum is quite a standard algorithm for clustering cytometry, mass cytometry data or high parameter flow data as well. But you can appreciate the expression profiles across the different clusters. So these are 60 clusters that were generated, and then we would annotate them based on canonical markers and into these meaningful cell clusters that we can interpret better. Here, just based on this clustering result, we can appreciate within the tumor microenvironment, the responders have much more immune cell infiltration. So really these light green towards yellow and orange bars, these are the lymphoid and the myeloid cells. A lot more already you can tell just based on the abundance level. There's more in the responder than the non-responder. But of course, this is just at the abundance. We can get the same type of data with suspension mass cytometry. And so really the unique aspect for imaging mass cytometry is get the spatial information. So we're analyzing the spatial information. There's so many different features out there. This is just one example of what we have done with this data. If you have all the cell-to-cell relationships based on their distances, and because there's so many of them within this data set, we want to distill this into trying to recognize which one might be the most important. And so based on response and non-response classification of these specimens, what we asked in this data set is what is the most important cell-cell distance relationship? And so using random forest and gene impurity statistic that comes out of it, the higher the statistic, the more important it is for classifying response versus non-response. And really the top relationships that we found were the relation between T cells, B cells, and then certain macrophage subtypes. So these yellow and brown bars showing at the top from a given T cell or a B cell, those are the most important relationships for determining response versus non-response. And interestingly, what we found was because we have 30 plus channels in imaging mass cytometry to profile these, we can then also use a lot of those markers to phenotype different subtypes of macrophages. As you might know, macrophages exist in a very large spectrum of phenotype. And so we want at least several markers to be able to phenotype well. And what we found was that close proximity with PD-L1 high macrophages actually is more indicative of response, interestingly. And then close proximity with arginase high macrophages are more indicative of non-response. So these are just the violent plots of the distances themselves that show this. And it helped us to interpret that perhaps, well, the proximity to PD-L1 high macrophage is something that we can overcome with nivolumab, anti-PD-1 therapy. And so that makes sense. Whereas non-responders, perhaps these arginase high macrophages are providing a mode of immune resistance that the current therapy cannot overcome. And so this is really helpful for us to gain that insight. Also within this core, we found an interesting correlate of immunotherapy responses increasingly appreciated as a marker of response in immunotherapy treated patients is lymphoid aggregates, so tertiary lymphoid aggregates or tertiary lymphoid structures if it's more structured. But using this terminology, TLAs, here you can appreciate that there are a lot more of these TLAs in the responders than the non-responder specimens. And using the bulk RNA-seq of the exact same specimens, what we have found based on the literature, they already established a 12 chemokine gene signature to then stratify patients, and this gene signature is indicative of TLA within these tumor specimens. So again, we were able to recapitulate that based on the literature that patients with high 12 chemokine signatures seem to do better. Of course, we're limited by the number of samples here, but they seem to do better than this low 12 chemokine signature patients. And we were using select set of specimens here. We used Visium to spatially validate this 12 chemokine gene signature. Of course, we're also looking at many other things within this large data set that we were able to generate using Visium. All of that is published in Genome Medicine last year. But this 12 chemokine signature is mapped onto the areas, exact areas where the TLAs exist. So we're able to validate the presence of this gene signature within the TLAs. And because they are associated with better response, we wanted to better understand, well, what are the phenotypes associated with this? Visium right now, each spot is about 100 micron, is probably the most commonly used spatial transcriptomic platform. Now we do have single cell resolution like Xenium platform that exists as well. But we wanted to understand the phenotypes of these immune aggregates with very high fidelity. And so we turned back again to imaging mass cytometry, designing yet another panel here, but now focused on phenotyping TLAs. And so this is just a representation here, and we were able to do additional analysis to understand how these TLAs look like in detail, and that's here at a preprint that we've also recently released. So in conjunction with all the work that we're doing in human, so we have all these hypotheses generation. How do we want to test that? Well, we want to test this in mice models. So we have studied different mouse models to understand what might be best representation of the human disease. And so we use imaging mass cytometry and suspension mass cytometry to, again, profile with enough parameters to characterize these the models so that we can better understand the biology. So I just wanted to hopefully communicate these main points. Mass cytometry, CyTOF, IMC are mid-range platforms for cost-effective discovery process with many different applications. And so I shared just some of them to you with you today. We can utilize multiple complementary data modalities, complementary in the sense of molecular scales, so RNA, protein, or spatial scales, small regions, and then immunohistochemistry, say, for whole slide validation and so forth. These type of approaches will add rigor and confidence in the discovery process. And again, utilizing both human and mouse models in parallel to have really a better handle on the mechanisms in play. So with that, thanks for listening. And I won't be able to stay throughout very much of the program due to another set of meetings I have to attend to right away, but I'll be happy to also answer questions by email and any other interactions that you would like.
Thank you, Dr. Ho. Then we can move to Dr. Have. And if anyone has any questions, just feel free to reach out to me, and I can direct your questions to Dr. Ho.
Thank you. Does everyone see my screen?
Yes. Mm-hmm.
Okay, so thanks, Dr. Ho. I think his presentation served as a nice segue to my presentation because he gave sort of a more basic overview of what imaging mass cytometry is, and my presentation focuses on the application of imaging mass cytometry in providing insight into the tumor biology in diffuse large B-cell lymphoma. So for everyone who doesn't know me, my name is Monirath. I'm a pathologist by training. I've been working in translational pathology, tissue-based biomarker validation and spatial biology for almost a decade. Currently serving as a director of pathology at MacroGenics, which is a clinical stage biopharmaceutical company focusing on discovering and developing monoclonal-based therapeutics for the treatment of cancer, as well as autoimmune disorders and infectious diseases. So before I start, here's my disclaimer. The opinions and scientific findings presented here today are solely those of mine and do not represent the views or positions of the company I'm working for, which is MacroGenics. So this morning I'll be presenting one of my postdoc studies looking at the tumor immune architecture of diffuse large B-cell lymphoma using imaging mass cytometry, which Dr. Ho just talked about. A bit of a background, diffuse large B-cell lymphoma, or DLBCL, is the most common subtype of non-Hodgkin's lymphomas, with about up to 40% of the patients having a refractory disease or developing relapse after the standard treatment with R-CHOP regimen. And unlike Hodgkin lymphoma, which responds very well to the PD-1, PD-L1 blockade, less than 10% of the relapsed refractory DLBCL responds to these checkpoint inhibitors, even though PD-L1 is highly expressed in a significant subset of the DLBCL and is associated with poor prognosis. So there's been increasing interest in developing novel immuno-oncology targets for relapsed refractory DLBCL. Many studies, including the one by Lens here, showed that the composition of the tumor microenvironment is a predictor of clinical outcome for the DLBCL. However, the complex immunophenotype of DLBCL's tumor microenvironment hasn't been characterized. So the aim of this study is to characterize the immunophenotypes and spatial architecture of the tumor immune microenvironment of 33 cases of DLBCL using imaging mass cytometry. So I think Dr. Ho gave a very nice basic overview of what the technology is. So that saves me some time. So another quick overview, faster than what Dr. Ho just mentioned. The workflow of imaging mass cytometry, or IMC, is very similar to the traditional immunohistochemistry, but instead of visualizing a few markers, 1 or 2, 3 markers at the same time, which in the case of immunohistochemistry, this makes big picture interpretation a little bit hard. And unlike flow cytometry, which enabled simultaneous detection of multiple markers at a time, but without any spatial context, as you can see, it's like lots of letters, lots of readout, but there's no spatial context whatsoever. A highly multiplex imaging platform like imaging mass cytometry allows us to simultaneously detect up to 50 protein or markers at the same time with intact spatial information of each cell, thus enabling high-level interrogation of disease biology. So we have both the marker information or protein expression and the spatial context of each cell in the microenvironment. I will skip this slide because Dr. Ho already explained what imaging mass cytometry is. So it's the same concept. We stain the tissue with metal label antibody, and we use the CyTOF or imaging CyTOF to ablate the tissue, resulting in readout of the ions, and then the readouts are rendered into pseudo-colored images that can be used for downstream single-cell segmentation, population quantification, protein quantification, and also spatial analysis. In this study, we stained 33 cases of DLBCL and a control tissue microarray just to confirm the performance of the antibodies that we use. And we use a panel of 32 antibodies as shown here on the right. Then using the IMC instrument, which is also the Hyperion, also called the Hyperion tissue imager, we acquired a 1 by 1 square millimeter region of interest on each sample. And the resulting multiplex images are then used for downstream single cell segmentation, classification, protein expression analysis, as well as spatial interaction analysis. In the coming slides, I'm going to highlight a few important findings from the study. In this slide, the bar graph on the far left here shows each sample in the study ranked by the proportion of tumor cells in red and the proportion of immune cell in blue. So samples on the top portions are those with high proportion of tumor and very low proportion of immune cells. In other words, they are called immune cold tumors. Whereas the samples on the bottom portion have the opposite proportion, meaning high proportion of immune and low proportion of tumor cells. So they are called immune hot tumors. And the second bar graph shows immune cell composition in each sample. But let's focus on the last two bar graphs on the right. They show the ratio of the tumor-associated or pro-tumor M2 macrophages and the ratio of the CD4 helper T cells to all immune cells in each case. So the analysis of this immune composition shows a negative relationship between the proportion of helper T cells and the tumor-associated M2 macrophages in immune cold versus immune hot tumors. The immune cold tumors have higher proportion of the pro-tumor or tumor-associated M2 macrophages and lower proportion of the helper T cells, while the immune hot tumors have higher proportion of helper T cells but lower proportion of the M2 macrophages. Also looked at checkpoint protein quantification. The checkpoint protein quantification on T cells reveals co-expressions of multiple checkpoint markers or proteins such as TIM3 and PD1, TIM3 and LAG3, as well as LAG3 and PD1 on the same T cells, suggesting that exhausted T cell phenotypes expressing multiple checkpoint proteins is a very common occurrence in relapsed refractory DLBCL. So with the XY coordinate of each cell known, there's a wide range of spatial analyses that can be performed in this study by measuring the distance from tumor cells to the nearest immune cells. We perform nearest neighbor analyses of tumor cells using an unsupervised clustering that results in 9 distinct tumor clusters, as you can see here, cluster A to G. So figure B here on the bottom left shows the average distance of those 9 tumor clusters to their nearest immune cell. Tumor cluster B, A, E, and G, for example, have the closest interaction with the surrounding immune cells shown by the shortest distance here. So we call them dispersed tumor region. Tumor cluster C, H, I, and F are located further away from the surrounding immune cells, so we call them tumor crust or the mantle regions. And last, the tumor cluster D is the furthest away from the surrounding immune cells, so we call them the tumor core. So it's in the center of the tumor, and that's why it's further away from the surrounding immune cells, which tend to surround the periphery of the tumor instead of infiltrating the tumor in the case of relapse refractory DLBCL. And figure D, shown here, shows the proportion of each tumor cluster across each case in the cohort. The last figure, figure E here, shows the interaction and repulsion between the different tumor regions and T helper T cells, represented by the blue line, cytotoxic T cell represented by the green line, and macrophages in magenta. So tumor cells in the dispersed region have the highest proportion of interaction with cytotoxic T cells, followed by helper T cells and macrophages, whereas the tumor cells in the mantle or crust region have no interaction with CD4 and CD8 T cells. The tumor cells in the innermost region or the core have no interaction with CD8 T cells and macrophages, but have some interaction with the CD4 T cells. With the distance from immune cells to various tumor regions known, we quantify the expression of CXCR3, which is a chemokine receptor expressed on effector T cells, or a T cell homing receptor on the surface of CD4 helper T cells in the different tumor region. And we were able to show that helper T cells with high expression of CXCR3, so here on the x-axis, this is the expression of CXCR3. So CD4 helper T cell with the highest expression of CXCR3 are the one that infiltrate the most into the tumor core, or those are the cells, the T cells that have the closest interaction with the tumor cells in the center or in the tumor core region, suggesting that enforcing the expression of this chemokine receptor CXCR3 on CAR T cells might be an attractive strategy to improve immune cell penetration into the tumor core region. So in summary, this study shows that immune cold tumors have higher proportion of the tumor-associated or pro-tumoral M2 macrophages, whereas immune hot tumor have higher proportion of the helper T cells. We also show that the presence of exhausted T cell phenotypes expressing multiple checkpoint markers is a very common occurrence in the relapsed refractory diffuse large B-cell lymphoma. And last, our nearest neighbor analysis revealed 3 distinct tumor regions with different functional states of immune cells. So we show that CXCR3 high T cell subset had the highest potential to infiltrate the tumor core, which can suggest that using or enforcing CXCR3 might be an attractive strategy to improve T cell infiltration into the tumor core if we enforce the expression of CXCR3 on CAR T cells. So yeah, that's it. I'm happy to take any questions if we still have time. Otherwise, I can take questions through email.
Thank you, Dr. Have any questions from the audience or folks online? All right, then. Well, thank you so much, our excellent speakers, Dr. Colantonio, Dr. Ho, and Dr. Have, for your amazing presentations. And please reach out to me if anyone have any questions for our virtual speakers. I also would like to acknowledge Frederick County Chamber of Commerce for helping us organize the event, and our sponsors, McBee Moore and Vanik law firm, for the great coffee and pastries. And thank you so much for coming. Please feel free to grab any bagels and food, coffee on the way out. So the next Biotech Connector will be on May 23, so stay tuned. Thank you so much.